I'm a PhD candidate in Dr. Eric Nelson's research group, and Anu's on my committee, and Anu and I have actually been working with this same source of commercially available vermicompost since 2005. So, but I'm going to be talking only about the disease suppression stuff today. And oh, I need the thing. I'm like, how do I get it? Okay. Okay. So, the overview for today, I'd like to give a little bit of background on suppressive composts, some background on my favorite pathogen in the world, which is Pythium, and some of the mechanisms of disease suppression. This is the major chunk of my PhD dissertation. And then also some more practical stuff. I've done some product development on liquid compost extracts, characterizing the process, something that's simple and can be done on farm, and some of the qualities of the end product. And then, like Meg said, I'd love to have some input on future directions, and definitely in the discussion here from people that have been using some of these materials. So, all right, let's see if all right, so suppressive composts. Obviously, this is not a new concept, and what's so interesting to me about it is just how long we have known that compost can help treat plant diseases. If you look at traditional agriculture, fragments of Cato's De Agricultura actually survived from ancient Rome, and he talks about the importance of, fermenting manure and using it. The 12th century Abu al-Jar's Kitab al-Falaha, which is an agricultural treatise, actually talks about if you have a sick tree, you should excavate around the roots, put fermented human manure, pack that around the roots, and then bury it back over. But we were joking in the car on the way here. He also says that to prevent hail damage in your orchard, you should put a naked virgin outside at night, lying under your tree, and that will protect your tree from hail damage. So you can't take everything from these, you gotta take it with a grain of salt. And then also the Aztec Chinampa system, which still survives today. There's been some work done with modern plant pathologists that have gone in, and they do maintain a Pythium suppressive soil by using different compost and kind of a muck silt system. So, and then of course in modern agriculture, you guys have probably all heard about Sir Albert Howard. He was a mentor to J.I. Rodale and really popularized this link between soil and plant health and compost. And then even in modern agricultural science, like mainstream agricultural science, suppressive soils were documented in the late 1800s, and the biological nature of this phenomenon was documented in 1959. So, okay, if we've known this for so long, why don't we still understand how it works? And trust me, it's not for lack of trying. There are a lot of people who have been working on this over the years. One of the complicating factors is that in 1 gram of soil or compost, there are probably 5,000 to 10,000 species of microbes, and 10 to the 8th cells. And of those microbes, we know how many are there because we can see them under the microscope. We can also assess them with different DNA-based technologies, maybe about 80% of them. A lot of them we can assess that way, but we don't know much about them because only 0.1 to 1 or 10% of them can we actually grow on a plate and find out what do they eat, what do they do, what things do they convert to what. So it's a really exciting time to be a microbial ecologist, I will say that. It's also a terrifying time as some of these new technologies come online. So with that, I'm going to jump right into Pythium pathogenesis. So we know that compost suppress disease, we don't understand how it works, and I would argue that this is a major impediment to using them effectively for disease control, because just as Meg has shown, sometimes things work, sometimes they don't work in different systems. So how can you really rely on that if you're a grower and you have to get some sort of crop to market? So Pythium, as I said, the best plant pathogen in the whole world. Okay, so all you need to know from this, again, like I can't be a plant pathologist without showing you a disease cycle. If you start over on the left, the vegetative hyphae give rise asexually to sporangia, which then germinate either directly by just growing a little germ tube or indirectly by producing zoospores. And the zoospores are what can cause disease, although these other methods can too. And the zoospores are unique. And again, Pythium is an oomycete, it's not a fungus. These are the water molds, which people thought they were fungi for a long time, but they're more closely related to algae. the neat thing about these zoospores is, can you click on that video? I think just maybe click anywhere on the slide. Oops, nope, not that one. With the mouse, yeah. Here, I can go back one. Go back, yeah. Oh, no, that's okay now.
Can you go backwards?
I'm trying. I'm going forward. That was me. Okay. So just click anywhere on the slide. Should.
Has to be on the next slide.
Okay.
Let's go back one again.
Whoops.
Yeah, let's go on.
Yeah, just.
Just go like
Okay.
this, all right?
It's going to be too small to see though. If you click on the black box, I tested it before I came in here.
You gotta start the slideshow over.
Yeah, you have to start the slideshow and then just click on the black box. Yeah, sorry.
No, no. Click with the mouse on the black.
Can you go backwards again? We are using the mouse to go on the back box.
Really? Okay. I do just have the video on the desktop if you want to play it. Sorry, I set this all up and then I tried to. So, there. Okay. So the unique thing about these zoospores is that here's some zoospores being produced inside a vesicle that's attached to a sporangium. You get about 20 zoospores per vesicle, and you see once they're released, they're motile and they swim. And not only do they swim, they actually swim preferentially towards, these are cucumber root border cells, and you can see on the left some zoospores actually attaching and starting to germinate there. And here you see that up close. So those are encysted zoospores who were able to find that root border cell by their nose. They don't really have noses, but you know what I mean. They can actually basically smell it, go towards it, and then infect your plant. So with that in mind, thank you. Sorry about the technical difficulty. Okay. So with that in mind, this is kind of a cartoon of that. So you have the zoospore who senses that gradient of exudates that are released from a seed, can find the seed, and then germinate and insist on that seed. And that process of directional swimming is called chemotaxis. Now, what happens if I inoculate cucumbers with zoospores and I grow them in sand? You can see that top row there is sand. The non-inoculated ones look fine. The inoculated ones are incredibly dead. And the numbers there are survival, percent survival, and then the letters are not significantly different. So if I grow them in compost that's been actually heat sterilized or pasteurized, I get a little more control than none at all, but it's not great. And then these three bottom ones here are grown in different batches of vermicompost, vermicomposted dairy manure actually, from a facility in Western New York. And we get consistently high levels of control. And again, I will give the caveat that this is, this is not field testing. This is just me in the lab getting a system that I can work in. But we were very excited to see this. So, and I will say a bit about this vermicomposted dairy manure. Like I said, Anna and I have been working with it since 2005, 2006. And we have results using it as an organic transplant media amendment. We have outreach videos and additional vermicomposters resources there. And I forgot to put that in my little packet. So you can also just Google Cornell and vermicompost and it usually comes up. So like I said, I wanted to set up an experimental system because I want to find out how this actually works. So I'm doing horrible things to these zoospores. I grow 10 cucumber seeds here and I inoculate the zoospores in the middle at that red X. And the zoospores actually swim up to 200 miles an hour. It's just, they're very small distances that they're covering. And so I'm basically making kind of a maze situation for them where I add them to the middle and then I try and find out, okay, well, were they able to reach this seed or not? And so when I actually plant the seeds into vermicompost, the seedlings are healthy and no zoospores arrive on the surface of the seeds. And the next thing I wanted to do was, okay, this is something my lab specializes in. because I told you about that complexity of, the 10,000 species per gram, instead of looking at the whole compost, I want to know what's going on the surface of the seed, because that is probably more likely to influence the pathogen than anything else. So I designed these experiments to look at the role of seed colonizing microbes. This is a little complex, but we have on the bottom a timeline here. I allow for an 8-hour period of seed colonization with the vermicompost. And then I inoculate here, I get an accumulation of zoospores, and then I allow some time for development of disease. So what that looks like in sand is if I plant a seed in sand for 8 hours, I transplant it against a sterile sand, and I add those zoospores, tons of them arrive. I actually am able to pull the seeds out after 24 hours and use molecular techniques to measure exactly how many zoospores got there. So I can say, okay, I have 22.9 femtograms of DNA per seed that is Pythium and phantom DNA. And I also know that it was colonized because none of them survived. And this looks very different with the vermicompost. So again, I plant the seed in vermicompost for 8 hours, but then I'm transplanting it to sterile sand. So now I have a situation where just the microbes that were able to colonize that seed within 8 hours are there. So I'm asking questions just about that community. And what we find is that after 24 hours, absolutely no Pythium. Well, a couple of them, there was, this is an average, there was like a little bit of detectable Pythium on there. But overall, basically none of the zoospores were able to reach the seed surface when it's coated with those microbes. And you see that reflected in the 90% seedling stand at 9 days. So this really got us thinking, okay, well, knowing what we know about the schematic with the zoospores and the seed, my hypothesis then was that seed colonizing microbes somehow are interrupting that zoospore chemotaxis. And they're probably doing it by modifying the seed exudates. And if that was true, my prediction was that the exudates here in kind of a different color, exudates from the seeds that are colonized with these vermicompost microbes, should actually attract fewer zoospores than exudates from non-treated seeds. So I went ahead and tested that too. So this is the same setup. I'm just using it to ask, how have the seed exudates changed? Like, what's going on here? So I do the same setup where I have half the seeds in sand that get transplanted to sand, the other half that get transplanted from vermicompost to sand. And then I basically just run water through that sand, filter it, freeze dry it, and then reconstitute it. And what I'm able to do with that is ask the zoospores what they think about it. So I developed these zoospore encystment assays where I have just a big petri dish full of swimming zoospores. I have auger plugs on a microscope slide that I infuse with those compounds that I collected from my system. And the slides are removed after 30 minutes, imaged, and then I count the encysted zoospores. And you can see that exudates from the seeds that were sown in sand and transplanted to sand had about 271 zoospores per, I think, 10 cubic micrometers or something like that. From vermicompost was actually no different than the water control. So whatever those seed colonizing microbes are doing, they're making it so that the chemical compounds that are released from the seeds are really not attracting the zoospores. And we kind of came up with two alternative and competing explanations for that. Well, the microbes are either eating something that the zoospores like and need to find the seed, so they're removing an important attractant, or the zoospores are pooping out something that the zoospores don't like. So they're producing some sort of a toxin. And so we did a couple of things to test that I don't have time to go into today, but we did find out that there was evidence of a toxin. So this is the exudate collected from seeds that were sown in sand. Those are healthy encysted zoospores that are germinating. And then this is the exudate collected from seeds with that 8-hour seed colonizing community from the vermicompost. And we actually see zoospore lysis. And this was very exciting. And we're following up on that a little bit now. But it's really our hope that by understanding this mechanism, we can create products and information for growers to use these materials more effectively. And with that, I'm going to jump right into the liquid compost extracts, which is a little more practical information. again, these non-aerated liquid extracts, I got into kind of the compost tea world when I got into grad school, and all the available information was talking about aerated extracts and adding nutrients and buying brewers and all this kind of stuff. But when I started to dig into scientific literature, there was actually some extensive work done in Germany in the 80s and 90s, not surprisingly done in Germany, because that is, of course, the home of Rudolf Steiner, and that is the home of the biodynamic method. So a lot of the scientists at that time were kind of influenced by those things going on. And the only problem with this is most of it's in German. So there's a reason why it wasn't kind of part of the popular information about these materials. So, and the literature shows that the non-aerated extracts are successful soil drenches, seed treatments, and foliar sprays in the greenhouse, and there's a lot of field efficacy data too. And the common recipe that we found was 1 part compost to 5 parts water. You agitate it, and then you just leave it in the bucket for 7 to 10 days. So we thought, okay, we're going to try and do that with the vermicompost. We're going to do a 1 to 5 and see what happens. And also some insights from the literature, some other things that I found that aren't part of kind of the mainstream discussion about these materials, is that there's actually been quite a bit of work done where someone took the same batch of compost, they made 1 aerated compost tea with it, 1 non-aerated compost tea, and then they tested the suppressiveness of both of them. And they found that aeration does not increase suppressiveness, and in some cases it actually decreases suppressiveness. And this definitely is not widely known, but it's pretty well established in the literature. And the total to active fungal to bacterial ratios, at least in the trial that Steve Sherrill and Walt Mahaffey did not actually predict which compost extracts would be suppressive or not, and they tested like 50 of them. And then this very interesting thing here, some require living microbes to be effective, but others do not. So all compost teas are not the same. Some may be working via microbes, and then some might be working by the chemicals that the microbes are producing, and you don't actually have to have a living microbe present. So with that said, we were interested to learn that Mark Elzinga and his head greenhouse manager, Roger, and I'm going to space on his last name, sorry Roger, they have been doing some really innovative things with a mixture of solid vermicompost and aerated extracts together for nutrient management. And they actually, those are their aerated compost extract brewers. They're using the vermicompost, and then the tubing there is they're actually, those are 500 gallons, they're piping it right into their irrigation system. But it's working for them, they love the system, but especially given the cost of that equipment, we wanted to develop something pretty quick and cheap. OFRF funded this. Okay, well, what can growers actually do that doesn't cost $50,000? And this is the same size for $250. Oh no, I lied. This is a 100-gallon tub, not a 500-gallon tub. But basically, we spent all the money on a sump pump. You can just get a bigger tub. And we did a 1 to 60 vermicompost to water ratio, and I'll tell you why. Like I said, those first things that I read were all saying you should have 1 part compost to 5 parts water. Well, if you look here at the electrical conductivity, it was 4. So that's 4 milliohms per centimeter or whatever, and that is really, really high salinity. So we had to tweak with the recipe. It was a big learning curve. The 1 to 5, they smelled horrible. They were like bubbling and fermenting, and they were incredibly phytotoxic. So we said, okay, let's back it off, and let's start diluting it out. And we definitely found some trends in how all of these different measurements respond. And here's an image of that. So here's just a phytotoxicity test with cucumber. That's cucumber and water. That was the 1 to 5 batch. Oops, not appropriate for the vermicompost. And then a 1 to 30. And really, it wasn't until we got to 1 to 60 that the root hairs looked like they were the same as in water. And so what I tell growers is if you're doing anything with compost extracts, test a range of ratios of compost to water, and you can do this phytotoxicity testing really easy. This is just cucumber seed and a little bit of like paper towel. And so you can kind of evaluate your own recipes that way, because this one killed the plants and the 1 to 60 protected them from Pythium. So there's a pretty big difference just based on that. And again, not a lot of the scientific literature had really characterized this whole process. And you see a lot of the popular information on compost teas saying, oh, well, the non-aerated ones are anaerobic. Well, I don't know if they're anaerobic, but they're definitely, there may be a tiny bit of oxygen here after day 1, but it's really, really small. So this is the days of extraction and then the average dissolved oxygen. And you can see it only took about 24 hours from that DO level to go from 6 to 0. And the spikes here are when the sump pump was going. So the sump pump was circulating it, but it wasn't actively aerating it. And then here you can see there's a little spike, but you really don't get much over like 0.3 parts per million. But I will say that in spite of that, the microbes were alive and well. I was able to sample from a couple feet down in that tub, and this is actually a piece of particulate organic matter that's just totally colonized with active microbes. That's a green stain. And what I found was about 10 to the 7th live bacteria per mil all over that 10-day period, even when the oxygen was down at basically 0. And that's actually an underestimate because clearly I couldn't count these. That was just a little too hard to count. And then up above I have what I noticed, and unfortunately wasn't really prepared to measure or count. I did notice a high proportion of actinomycetes, which are filamentous bacteria, who are associated with Pythium suppression, but I have no idea if they were actually involved in this case. So here are my extract suppression results. Again, the same kind of thing where on the left it's non-inoculated, on the right it's inoculated. I got obviously disease in sand. I filter sterilized the extracts, and in this case I needed the living microbes to get that suppression. They really weren't effective when they were filter sterilized. And then I have the fresh non-aerated vermicompost extract. And then we were also messing around with, well, what if we freeze dry that and then reconstitute it? Is it still suppressive? And it was. Something that I was really shocked to find, I kept testing it just for kicks. I had a bucket of it, and I would pull it out and test it every once in a while. And I had the same level of suppression for up to 60 days in storage at room temperature. And that's something that with the aerated extracts, even the people selling them will say you have to use it within 4 hours. So in my mind, for practicability, I'm pretty impressed with the fact that I can get the same level of Pythium suppression 60 days later with it sitting on the lab bench. So that was exciting. So future directions, I'm right now working with Donna Gibson at Cornell, the USDA ARS. She's a natural products chemist that does a lot of work with biopesticides. And we're trying to figure out what that toxin is. Is it something that we already know about? Is it a new one? We don't know. We'd like to screen different species of zoospore-producing oomycetes against that toxin to see if it's effective. Because if it's something that's effective for Pythium and Phytophthora and Aphanomyces, that would be fantastic. And then we also eventually would like to determine which microbes growing on the seed surface are actually producing the toxin. And I'm doing just some practical stuff right now. We're looking at different ways to use both the liquid and powdered solid vermicompost as a seed treatment to see if it would be an effective seed treatment. Now here's the issue, again with the EPA regulations, this could never be registered as a biopesticide because it has a complex group of microbes. So if we can find an effective kind of seed treatment, it would be something that we could encourage growers to use as a cultural practice, but it's never anything that could ever be marketed as Pythium suppressive or suppressive on any disease. And that's one of the reasons why there's so few people looking at these materials, because if you can't sell it to someone as suppressive, why would you spend years trying to figure out, like, which I ask myself that every day. So really my take home is by understanding the mechanisms of suppression, hopefully we can develop more consistently effective products and practices. And I would like to point out that Neil Mattson in Cornell Horticulture, he just got a grant with our industry collaborator, same industry collaborator, Worm Power, to look at organic nutrient management of the solid vermicompost and the extracts. So they're just starting up that project right now. And with that, I'd like to thank all the funders and everybody involved. Thank you so much.
Food safety.
Yes.
They're not letting us spray compost teas, whether they're air extracted, non-aerobic. What's your answer to that?
I'm glad you mentioned that. So I'm not up with a lot. I actually did a human pathogens and compost tea project for my master's, so I was following all the news on that a lot better, and I've been a little bit out of that world. My news on that is that's why I'm working on it as a soil drench, because it's a little bit complicated to ensure the safety, because this is manure in a bucket. Yeah, it's composted manure. I didn't look at safety, but I know that there's a lot of work on human pathogens on vegetable surfaces. I thought that if you had a compost that passed an E. coli kind of test, that you could make it yourself on farm. Is that right or not right? Like what are the current NOP?
No, I think it's can't.
It's true compost.
We're looking
It's
at
not-
you.
The question is it compost or manure? Right.
Right. But if it's compost that's OMRI listed, because I know this vermicompost has to go through all of the pathogen testing and everything to be OMRI listed. It was my understanding that people could use it, make it on farm if it passed those tests, no? And like if you use the same batch of compost.
I mean, we don't really have a rule about it. In fact, we purposely left it out of the last guidance on compost because there's
Okay.
so much different information about it. But what I think Elizabeth is talking about is the upcoming food safety regulations, which we haven't seen yet. Some people may know more about than others. So.
And these are just certified organic, or
No,
it's
no, Food
safety
Safety
across the board?
Modernization Act.
Okay.
Across the board, everybody. So
Well,
does anyone have any
luckily I work with
knowledge?
seed pathogens, and they could be an effective tool at that point, and in that way they're not on any edible part of the plant, but it's not crazy to think that there could be a problem with food safety.
Another question? Over here? Luke?
How do you characterize the vermicompost? How do you know? Is it all the same? Are there any majors of how it might be good or bad?
How do you characterize compost? Well, I will say that Jean Bonatal at Cornell Waste Management Institute is giving a talk this weekend on compost quality indicators and how you can produce things to make how you can make high quality compost on farm. One of the reasons that we were excited to work with this particular commercially available compost is because it's made indoors, it's highly process controlled, but yeah, compost quality is kind of like soil quality, again with 10,000 species of microbes, and we don't know what most of them are doing. We look at nutrients, we look at electrical conductivity. A lot of growers say they like the vermicompost because you can put it in that automated equipment. That's why I showed the seeding trays at Elzinga Hoxema, because I can put this in my equipment and it doesn't gum it up. It's like friable. But I don't know, Anu, do you want to attack that one? Compost quality?
Well, were you asking about vermicompost?
Yeah,
specifically?
vermicompost.
Are all worms created equal?
No, and not all facilities are created equal either. And that's another interesting one where someone can put so I'm in the very beginnings of trying to start an industry association for vermicomposters, because the U.S. Composting Council, we're kind of like the ugly cousin. They're like, yeah, we don't want to deal with you at all. It's like different, it's weird, we don't know. And right now, there's no minimum stocking requirement. Like, you can put 2 worms in a windrow and call that vermicompost. So, yeah. Get back to me in 10 years. I'll see what I can do.
One more question, then we'll bring Meg back up as well and have more discussion.
Are worms livestock? Just kidding.
They are. Actually, that's a fantastic question. Are worms livestock? So Tom Hurley, who's our industry collaborator, who's the president of Worm Power, he went through machinations like you can't believe. He wanted to get a value-added producer grant, and they said, well, you're not eligible because you're a composter. He said, no. like the 2007 Ag Census, right here under other, next to bull semen, is earthworms. So I am a livestock producer, and not only did he qualify for the grant, he got the grant. So yeah, 2007 Ag Census, go. So yes,
There it is.
they are livestock.